My guest today is Professor Stephanie Cherqui, who is Professor of Pediatrics at the University of California, San Diego. Her research project is focused on the molecular characterization of cystinosis, a metabolic hereditary disease, and gene therapy. Welcome, Stephanie.
Well, thank you very much for inviting me. It's a pleasure to talk to you today.
Yeah, thanks for doing this. So, obviously, I don't know much about cystinosis. I gather it's a rare disease. Incidence rates in the range of 1 to 100,000 or
That's
something along
correct.
those lines. So really rare disease. It is a genetic disorder. And so you have to have fathers and mothers gene disorders sort of come together so that both of that particular gene has a problem for the disease to appear. So you say here, you have a number of papers in this area. I know that you have been doing research in this area for a long time. Cystinosis is an autosomal recessive metabolic disease that belongs to the family of lysosomal storage disorders. And so the little I understand about this is that there are materials inside the cells that, for whatever reason, because of the gene disorder, the cells cannot get rid of. So you have an accumulation problem in the cells, right? So what exactly is cystinosis?
Yeah. So actually, yes, I have been involved in cystinosis for a very long time. I did actually my PhD in cystinosis, and we actually identify the gene involved into this disease. And so the gene called CTNS encode a lysosomal transmembrane transporter of cysteine. So
Yeah.
this allows the cysteine, the amino acid cysteine, to exit the lysosome. So the lysosome is kind of a recycled bin of the cells, and this is where the protein are enter the lysosome, and they are cut in many amino acids. And the cysteine has to exit the lysosome to be reused by the cells. And cystinosine, the transporter of cysteine, is missing or non-functional in the patient with cystinosis. The issue
Yeah.
is that.
So
Sorry.
sorry to interrupt, Stephanie. So let me see if I can understand this. So the lysosome is inside the cell,
Yeah.
and cysteine is a protein
Cysteine is
that is
one of
in the
the,
lysosome.
it's
Yeah.
an amino acid. So it's like one of the breakdown from the protein. So the protein
Okay.
contains cysteine. And so when the protein enters the lysosome to be recycled, it's break down by, several amino acids. Actually, cysteine is two cysteine together,
Yeah.
and it require a transporter to be able to exit the lysosome.
So the cell is, if I understand this, purely from a mechanical perspective, the lysosome is sort of a manufacturer of cysteine. And in a normal cell, there is a transport mechanism. It can take
Mhm.
that out and
Yeah.
into the bloodstream. So I have two questions. Why do we need? Why does the body need cysteine? What is the function?
So cysteine is part of a protein. So every protein in our body requires cysteine. So it's like arginine or any other amino acid. So it's required for the protein. Actually, cysteine is a two cysteine together, and when it's exit the lysosome, when it's going to the cytoplasm, it's break down into two cysteine, and it's reused by the cells to make proteins.
So it's almost like a starting material in the
Yes,
pharmaceutical
it's
process.
part of it's like a building block, that you need to make a house or something.
So,
But the issue is that CTNS is expressed in every single tissue in our body. So when in the patient who have a mutation in CTNS, so who have cystinosis, actually accumulate cysteine in all the organs. So the lysosome become dysfunctional. So the cells becomes dysfunctional and start to create the cysteine crystals in the lysosome that are characteristic of this building of cysteine storage. And eventually the cells die. So little by little, progressively, all the organs in the body fell in the patient with cystinosis. And so this start by the kidney. So they are the first organ to be affected for cystinosis. It's not fully understood why it's the first organ, but it's so this start in infancy with dysfunctional kidney, and eventually the patient have end-stage renal failure and require kidney transplantation. But this patient also accumulates cystine crystals in the cornea, which leads to photophobia and eventually blindness. They also have endocrine dysfunction, myopathy, so muscle weakness that also lead to their death. They have neurological defect, etc. You name it. It's like this patient, unfortunately, have very profound organ defect and poor quality of life.
Yeah, so it's a really bad disease in many ways. But going back to the mechanics of this, so in a normal cell, there is a transport mechanism. We call this here proton-driven cystine transporter. So in a normal cell, it can basically take it out of the lysosome and put that into the bloodstream. When you have this problem, you don't have that transporter. So essentially, things are getting accumulated, right, in the lysosome. Yeah. It just sits there. Yeah.
Yeah, and it doesn't take it into the bloodstream. It takes it from the lysosome to the cytoplasm, where it's reused for the cell homeostasis. And yes, because the transporter is missing, then the cystine is stuck within the lysosome and accumulate.
Yeah, I can't even imagine what this situation. I mean, I know that there are multiple types of cystinosis. There's early onset, late onset, and like just ocular cystinosis. But if it is accumulating in the cells, that is not a sustainable process, is it? I mean, what happens? They get it crystallizes, and
yes.
What goes on? Yeah.
Yeah. So the cystine that accumulate crystallizes. So that's why all the patients have this very characteristic accumulation of cystine crystals, which actually help for the diagnosis. Because if there is a suspicion of cystinosis, they usually try to see, have cystine crystals, for example, in the cornea, and see if they have crystals, then we can have a diagnosis of cystinosis.
Yeah, so the crystallization of this is so sort of a systemic problem. It's happening everywhere. Yeah. And so there could be a lot of different organs ultimately involved. But there's a specific variety that affects the eye more than the other organs.
So actually, it affects the kidney more than the other organs. But again, it's not completely understood why the kidney are the first affected, and all the patients require kidney transplantation. Then the second tissue to be affected is indeed the eye, with abundant accumulation of crystals within the cornea.
So, so I know that the uric acid, for instance, crystallizes and it gives people gout. But this is very different from that, right? This is a systemic, every cell is involved in this process, right?
Yeah. It's very different. Actually, some people mistake this, cystinosis with uremia, but it's not the same thing at all. And the accumulation of crystals is inside the cells, and which make it very a critical disease, because with this accumulation, the cell die, and eventually the organ.
So a lot of your focus has been gene therapy. So essentially, that transporter is missing. The ideal situation would be to replace a transporter on every cell. It's easier said than done, right? I mean, I don't even know how do you go about doing that.
I wouldn't have known either. I mean, actually, actually, I wanted to do a gene therapy very early on, and for my PhD already, which was too early to do a PhD on gene therapy. But I started to work on cystinosis, and I was and I thought it would be very nice to go from the gene to gene therapy. So I started to work on cystinosis with this in mind. But when we found the gene, we first realized it was expressed in every single tissue. We second realized that it was a non-secreted protein with a transporter. That was the worst disease model to do gene therapy on. So yeah, I wouldn't have chosen this disease at all if I would have to go back. But actually, I'm glad I did, because after when I became a faculty and I knew the tools for gene therapy, I was already involved a lot with the family and the community with cystinosis, and it was now a personal fight that I really wanted to find a better treatment for these patients. So, of course, how do you bring a gene everywhere, especially when it's not secreted? So you really have to target the most cells possible. So that's why I turned to the ex vivo gene therapy in contrast to in vivo gene therapy. The in vivo gene therapy is that when you inject into the body a viral vector containing the missing gene. So, and most of the time you use adeno-associated virus. The issue with in vivo gene therapy is that you are very constrained with a dose, and it's impossible to target all the tissue with in vivo gene therapy. So, so that wouldn't be okay for this kind of disease where you have to really bring the healthy protein in most of the tissues. So I chose then to go to the ex vivo gene therapy, meaning that we are using the stem cells to that we gene correct or gene modify ex vivo before we put them back into the body. Yeah. And actually, there were two, the bone marrow stem cells that we have in our, all of us in our bone marrow, have really the ability to be able to target every single tissue in our body if we have any kind of injury. So I was thinking, maybe because in cystinosis patient, all the tissue are damaged. So we the cells might be able to engraft into this tissue. And so we have two main bone marrow stem cells in our bone marrow, the mesenchymal stem cells that make, tissue cells, and the hematopoietic stem cells that give rise to blood cells. So I really believe that the mesenchymal stem cells would be the one that would really make the difference, because we want to target tissue cells. And I use the hematopoietic stem cells as a negative control. But against all odds, I realized that, so the mesenchymal stem cells had only transient beneficial effect, because now we know that they cannot engraft long-term in tissues. But unexpectedly, the hematopoietic stem cells had a very dramatic impact on the disease. So first, we found a lot of hematopoietic stem cell derived cells that engraft into all the organs, and we realized that it led to a dramatic decrease of the cystine content in all these organs, and a long-term preservation of these tissues, of the kidney, the thyroid, the eye, etc. So that was a very amazing and unexpected results. And again, because the protein is non-secreted. So what we realized there was that actually the hematopoietic stem cells, what they become is that because they give rise to a lot of hematopoietic cells, the blood cells, but the monocytes in the blood cells have the ability to become macrophage into the organs, or macroglia into the brain. And we realize that the macrophages, because they become resident macrophage, so they engraft long-term into the tissues, they have the ability to transfer the lysosome via tunneling nanotubes. So the tunneling nanotubes are this long tubular protrusion that can make a bridge between cells, so connection, and deliver the lysosome carrying the cystinosin protein in this case. So that was a kind of a novel mechanism of action to explain how hematopoietic stem cells could rescue disease like cystinosis. And so this is what the innovation of my lab.
So I don't quite understand it, Stephanie. So in some sense, you need some sort of a messenger to remedy the issue in almost every cell in the body. And stem cells, some type of stem cells appear to do that in this case. But is that a sort of a platform technology in the sense that if there's a systemic genetic disorder, could you use the same idea for others, for other
Sure.
diseases?
You are completely right. So now what happened is because we use hematopoietic stem cells in disease that was not thought to be able to work, we were able to make intellectual property on that kind of disease. And so it was like the lysosomal disease due to a transmembrane lysosomal protein. And so we were able to show that this works also in another lysosomal disease called Danon disease with my collaborator, Eric Adler. He works on this disease for many years. He's a cardiologist. And actually, the patient with Danon require a heart transplant. They also have eye defect and neurological defect. And the protein involved is a LAMP2B protein, which is also a transmembrane lysosomal protein. And so when he saw me one day, presenting at UCSD, he's also from UCSD, he said, "Hey, wait a second, why don't we try on Danon?" And sure enough, so we tried and we were able to rescue the disease. And we did the same thing then for cystinosis. So we developed the ex vivo gene therapy approach, and we also now starting the safety study for one day starting a clinical trial for Danon.
Yeah, I would imagine the mechanics of this is quite complicated and
Yeah.
potentially dangerous.
Yeah.
But it's sort of swapping out existing bone marrow in some way with replacement with a genetic modification, right? Is that the
Yeah, and that's an important point. So, if with this kind of results, you would think, okay, so why don't we do a bone marrow transplant? Actually, a bone marrow transplant, when you take stem cells from a donor, a foreign donor, there are high risk of mortality and morbidity associated with that, more than 20% of risk because of this graft versus host disease. You are putting cells that are not yours, so oftentimes they will react against your body and you can die from that. Actually, one patient who was 16 years old has been transplanted with allo transplant, so using foreign hematopoietic stem cells in Belgium based on our work. And unfortunately, the patient died from these processes. They still saw benefits from the transplanted hematopoietic stem cells for cystinosis disease, but the patient died from graft versus host disease. So it is very important to understand that you cannot do bone marrow transplant in any patient. It's very risky. So what we have to do, and what is much safer to do, is to actually take the patient own bone marrow stem cells and hematopoietic stem cells and to gene modify them or gene correct them ex vivo, and then we can transplant them back by doing a bone marrow transplant. So this requires a short-term myeloablation, so using a short-term chemotherapy to remove the patient's own bone marrow stem cells, so this makes space into their bone marrow. And then when we infuse the gene-modified hematopoietic stem cells, they will go to the bone marrow by themselves, reside there, multiply there, and for the rest of the life of a patient. So now we have a reservoir of healthy cells for the rest of the life of a patient. And as we've shown is that when it's required because of tissue damage, they will travel to the tissue, engraft, and provide the healthy protein to the disease cells. So that's kind of a really self-regulated one-time treatment that is very powerful, actually.
So there's no rejection risk if you do
And
that.
there is no rejection risk, except if for any reason the patient or the body can react against the new protein that you are adding. So there are also safety studies that you always have to verify, but it's more rare. But you always have to verify that. But in the case of cystinosis, for instance, there is no issue on immune reaction against the cystinosin protein for patients who don't have it. But there is no risk of rejection from the stem cells themselves.
Yeah, so if I understand this correctly, Stephanie, so you take part of the bone marrow of the patient and you modify it with the vector, and you don't take all of it, right? You take some bits of it and then you grow it. Do you grow?
Yeah. So what we do exactly is that we mobilize the hematopoietic stem cells. So we call that the CD34 hematopoietic stem cells. So it's the hematopoietic stem and progenitor cells. So we use so we first mobilize them in the bloodstream, and we do what we call an apheresis. So it's a machine that will take the white blood cells and send back the red blood cells into your blood, so that you can take much more. And from this apheresis bag, we isolate the CD34 hematopoietic stem cells. So this has to be done in what we call a good manufacturing practice facility. And so this for cystinosis, it was done at the GMP facility at UCLA, directed by Donald Cohn. And this is where the gene modification of the cells were done before and after characterized, make sure they are safe to transplant, and then they were able to be transplanted and reinfused into the patients.
Reinfused in the patient. So the chemotherapy aspect of this is essentially removing what is there by chemotherapy?
Yes, it's to remove your own bone marrow stem cells.
Yeah.
So like that, the one that have not been gene corrected, they will be removed. It has to be, otherwise the new stem cells are not engrafting into the bone marrow.
Right. Yeah, I mean, it's a big process. And in the middle, the patient is at risk from almost the immune system, I would imagine, is sleeping.
Yeah, I mean, we completely replace. So, I mean, so the risk after, after this kind of bone marrow transplant, the patient has to stay at least 3 to 4 weeks in the hospital, because there is the risk that exists is that they are more prone to get infection and do not have their immune cells to fight it. So they have to be in a secure place, in the hospital, to where, but bone marrow transplant is happening every day, for many disorders, like, cancer, etc. So, people are used to doing that as in a very safe environment.
Yeah. I mean, there are no options, it sounds to me. I know there's a chemical agent that is more symptomatic, isn't it? I mean, it's not a cure.
So what are you doing? The chemotherapy?
Yeah, what is it called? There is a medicine that's used for the disease, right? A chemical agent.
Oh, sorry. Yeah. So the drug that currently exists for cystinosis is called cysteamine, and it's a chemical that enters the lysosome and is able to break down the cysteine, and so after the cysteine is able to exit the lysosome. But it's just delay the progression of the disease. So it's very important, because before the patients were dying very early from the disease. Now they can live until young adult. But still, they still develop the complication. They still have a poor quality of life. But with this drug, they are really able to live longer. But clearly, it's a need of better treatment. And that's the reality for most of the genetic disorder. Very often, they lead to profound tissue damage and poor quality of life. And most of the treatment cannot be addressed by a regular medication. And this is when why I think, gene therapy is really the future of medicine for this kind of disorder. And we see that more and more now because before it was considered science fiction. Now we have more and more FDA approved gene therapy product for genetic disorder. And we see a very dramatic impact of this gene therapy product compared to regular treatment.
Yeah, we don't know anything about this, Stephanie. So I was wondering if it is purely genetic disorder, why are we seeing late onset of the disease? Shouldn't everything be early?
No, that's a very good question. So there are different kind of mutation in the case of cystinosis that we can find in the cystinosis gene. And we have done, so during my PhD, we have done some functional assays that determine the impact of a mutation in the protein. And what we've shown is that in the infantile form of a disease, most of the mutation lead to the non-functional protein, where there is no transport of cysteine at all. In the later form of a disease, we can measure some residual transport of cysteine. And so that's why the phenotype is less important.
Yeah, so I get the mutations that we're seeing. I don't remember the name of the CNT something. What was the
CTNS?
Which one?
The gene? CTNS?
Yeah.
Yes.
Yeah. And so I guess there's a gradation there in terms of, so it's not a pure case, is it? I mean, so you could have some small amounts of transportation going on,
Yes.
even though you have the mutation.
So for example, in the juvenile form, we have seen some residual transport. There is also an ocular form of cystinosis that is found in adult. And in that case, we found a much higher level of transport of cysteine. So, it depends on how much cysteine can still be transported, and that determines the phenotype.
And so if I understand this correctly, Stephanie, so this is sort of a starting material for a lot of protein production. If it is not getting there, then a variety of other issues are going to happen to the body, right?
For cystinosis?
Yeah.
Yes, I mean, that's why the patient have kidney failure, blindness, muscle weakness, endocrine dysfunction. So yeah, I mean, there is a lot of defect in each organs that leads to the organ failure.
Yeah. I know that you have another paper here, which is sort of on the same lines. So Friedreich's ataxia.
Mm-hmm.
So what is that? It is also an autosomal recessive neurodegenerative disease. So what's happening there?
So why we started to work on Friedreich ataxia. So first of all, I was not at all familiar. I mean, I heard about this disease because when I was in PhD, I was working with a colleague who had a son with Friedreich ataxia. So that's why that's the first one that came to my mind when we wanted to work on mitochondrial disease. So why I got interested in mitochondrial disease is because the tunneling nanotube that can transport the lysosome can also transport mitochondria. So I was thinking, if we can make a difference in lysosomal transporter, because the organelles can be transported or transferred, maybe we can do the same in mitochondrial disease. And so again, this was novel and in the field of gene therapy for mitochondrial disease. And I was like, okay, let's try to make a proof of concept. So again, because I knew my colleague had a son with this disease, and this is also a terrible disease where the patient are born, normal, and suddenly they start to have locomotor deficiency, and they get this diagnosis of Friedreich ataxia, and it's really a death sentence, where the patient will progressively lose his locomotor ability, will end up in a wheelchair, and for years, and end up dying from cardiac dysfunction. So it's very terrible. And despite of the fact that many people work on many groups, many investigate or work on this disease for many years, and there is no treatment that can really stop the disease. So instead of going after the neurons or, trying because it's neurodegenerative, everybody thinks, okay, we have to fix the neurons.
Yeah.
I was thinking maybe let's go to another approach where, we know, because what we've learned and what others have shown is that the hematopoietic stem cells can become macroglia-like cells in the brain. And these macroglia they are really important for the homeostasis of the neurons. And they are able to transfer a lot of things to neurons. So I was thinking maybe they can also transfer the missing protein here. So we did the proof of concept in the mice with Friedreich ataxia, and it was really like a dramatic impact. I mean, I always remember when I was with my postdoc and we went for the first time testing the mice with, you put the mice in some behavior testing, some machine that can, you can see how they react. And we could tell which one had received the wild-type hematopoietic stem cells as opposed to the one that received the disease cells. It is really black and white. I mean, it was really a huge impact. And it was really amazing moment. And so we knew it was working, but after we're like, okay, now we have to understand why. And actually, the primary defect is the death of neurons in the dorsal root ganglia, which I didn't even know where it was at the time. So we had to learn. After which we realized is that actually, the hematopoietic stem cells were becoming macroglia-like cells in the CNS, dorsal root ganglia, and they were able to transfer the frataxin protein to the neurons and to prevent the neuronal death. And so that was very important findings. And so we took the disease from a completely another angle. And now we have received, found from the CIRM, California Institute of Regenerative Medicine, and FARA, the advocacy group, to really move forward this treatment to a clinical trial. So we are now at the stage of safety studies. So we are really making great progress. We will have our first interaction with the FDA early next month to know exactly what is required to get to a clinical trial. So we are very excited by this work too.
Yeah, so from a layman's perspective, Stephanie, I'm thinking this appears to be sort of a very broad application. If I understand this correctly, you take some type of stem cells, you modify them, and you modify them in such a way that it has a systemic effect on the body. And there could be many diseases that this could influence. You have another paper here in Alzheimer's disease, which is a little different. You say here stem and progenitor cell transportation leads to complete preservation of neurocognitive performance and partial preservation of blood-brain barrier integrity. So what is the concept here in Alzheimer's?
So again, from cystinosis, a rare disease, we went to Friedreich ataxia, another rare disease, but less rare than cystinosis. And we learned so much from cystinosis with the mechanism of action that we were able to apply that to Friedreich ataxia. From Friedreich ataxia, we learned the power of hematopoietic stem cells, and others have shown that also for metachromatic leukodystrophy, adrenoleukodystrophy. So we know, and we are in clinical trials, so we know this is translatable to human. So we were, and after in Alzheimer, it's like the cause of Alzheimer is still not really well understood. Some people say that it's because of the plaque accumulation. Some people say it's because of a blood-brain barrier. Some people, everybody has their own hypothesis. But some people were saying that the macroglia cells, actually their neuroinflammation macroglia cells might be involved in the pathogenesis and the neurodegeneration. So we're thinking if this is true, then if we use healthy hematopoietic stem cells, maybe we can stop the progression of Alzheimer's. So that was the rationale behind this work. So again, we tested that in the mice. We tested the mice into this, behavioral testing, and again, the results was really beyond our expectation when we could completely rescue the memory function or preserve the neurocognitive function of the mice with the memory, the sense of danger, etc., was completely preserved in these mice. And what we've shown is actually that the neuroinflammation was very reduced. I mean, it was non-existent. Whereas in the mice with Alzheimer, there was this dramatic neuroinflammation, and the macroglia cells there were so inflamed and activated that they have a potential to eat the neurons, so to lead to neurodegeneration. So by using the hematopoietic stem cell, we completely stop that. And we believe that this is really a big part of the pathogenesis. So obviously now we have two more direction on this work. Is like we want to develop for the familial AD a more gene therapy, ex vivo gene therapy approach, like we have done for cystinosis or Friedreich ataxia. But we also want to understand better, how the hematopoietic stem cells completely stop the progression of Alzheimer. And I think this is very important for understanding the disease and finding drug targets.
So if I understand this correctly, so microglia inflammation, if that is what's causing it, it's a little bit of a runaway process, right? So it gets inflamed and it creates more inflammation, and then things go down down the drain from there. And so, are you saying that you're stopping inflammation, or are you saying that you're actually replacing some of the cells?
So we are replacing the macroglia by macroglia-like cells, and we are preventing the inflammation. Now there are some response to that we still have to figure out. If the inflammation is already very intense, can we regress that? I mean, reduce it? I mean, there are some things we still have to understand. But in that case, by replacing the macroglia by healthy one, we completely prevent the inflammation and the activation of the macroglia-like cells. And we show that we stop completely the progression of the disease itself.
So, like you said, there were a lot of hypotheses around the cause and progression of Alzheimer's disease. But if with this technique, you're actually demonstrating improvement in neurocognitive performance, then it should be sort of pointing in this direction, right? That is sort of the cause of it.
I would say that, but there are many people who would say it differently. I never saw a disease where nobody agree with each other. So, I think that's what the beauty of this. I'm very excited by this project because there's so much to learn. So we are really making a big effort now to understand this process because there is so much that is unknown, and that I think we have a perfect model to understand it.
So we look forward in conclusion, Stephanie. So where do you think this research will go? What areas are you really excited about, and what do you want to do in the next 5 years?
So I'm really excited by the potential of ex vivo gene therapy. As you said, now we have a platform. I really believe this is very powerful, and there are many disorders that we can rescue with this technology. So this is what I really want to develop. But I also want to develop gene therapy in general. I also have some project when it's in vivo gene therapy, but for some very specific tissues. I'm happy to see how gene therapy now is considered as part of medicine. I have a course on gene therapy and I remember that I started that 6 years ago, and I had only one. It's for medical student and graduate student, and also pharmacy student, and I had only one medical student. Now it's 50% of the students are from a medical field. So I think now even the physician really consider that also part of a medicine. So for me, it's a big victory. I also now I just recently launched the gene therapy initiative at UCSD with philanthropic funds. And the goal of that is to really help people to develop gene therapy. There are many people who work on disease and have the perfect disease model for gene therapy, but they don't know maybe how to do it, or they don't have a fund to start, this kind of project. And so this is what we want to do. So I really want to support the development of gene therapy in general.
Would you say from a medical practitioner's perspective, in vivo, they can understand it, they can manage it. Ex vivo is a process, isn't it? I mean, it's a
we can manage both. I mean,
yeah.
So there are many clinical trials with in vivo gene therapy and many examples, but ex vivo too. For example, so for cystinosis, we developed that here, and all the physicians got on board, and they are doing a great job. But there are many other examples, as I said, metachromatic leukodystrophy, adrenal leukodystrophy, sickle cell disease, beta thalassemia. I mean, there are many of them. Yes, it's a process. Yes, we have to put in place, but at the end of the day, it seems to be a very safe approach. And so I truly believe that there's so much to learn about that and the power of it, that it will become much more applicable and used in medicine.
Yeah. Excellent. Yeah, good luck with everything that you're doing. I think it is very important work. And if we can go in and modify cells and genes, we can cure a lot of diseases, I think. So this is
It's not
this is
all, but we can at least try to do a lot of them.
Excellent. Yeah, thanks for spending time with me, Stephanie.
Thank you very much. Bye-bye.
Thank you.